My name is Jacek Mokrosinski. I'm a research associate at the University of Cambridge, Metabolic Research Laboratories. I work with Professor Sadaf Farooqi. I'm a postdoctoral fellow. Our group is focused on studying obesity. Specifically, we're looking at the genetic factors which determine body weight regulation. I'm a pharmacologist by my training, and I'm particularly interested in cell surface receptors. I'm especially focused on G-protein coupled receptors. It's the largest family of membrane proteins in the human genome. And the one GPCR that I'm particularly interested in at the moment is called Melanocortin 4 receptor. So we want to learn how our brain, how our body can influence our body weight. And MC4R receptor is a key player in this whole mechanism, in this whole system. So studying both severely obese patients and studying people from general populations like UK Biobank and others inform us more about the actual mechanisms governing these processes. The primary tool which sourced from Promega is called NanoBiT. It's a system which allowed us to study the function of melanocortin 4 receptor in great detail. We were able to apply this method, this system, to study beta-arrestin recruitment by melanocortin 4 receptor. NanoBiT system relies on two fragments of nanoluciferase enzyme. These two fragments are inactive each individually, but when combined together, when complemented together, they form fully active enzyme. The small fragment and the large fragment are attached, are fused with proteins of interest. In our case, it's melanocortin 4 receptor and beta-arrestin, and if they are expressed individually, they don't give us any readout. However, if we stimulate the cells in which we express these two fragments, if two components of the system complement and nanoluciferase can be formed again as an active enzyme, we're detecting really strong light. NanoBiT system is brilliant for applying in GPCR research because of the size of the nanoluciferase enzyme. It's a very small protein, and in any assays which are used to study G-protein coupled receptor signaling, you're very dependent on the modifications you do to the receptors. Smaller the tag, smaller the probe you can attach to this receptor, the better, because the size matters here, and whatever you attach to the receptor, it might influence how it works. Simply, the receptors are small themselves, so they need very, very small probes, and nanoluciferase does that. We run our assays in real time in live cells, so we measure changes and we measure the signaling over time as we stimulate the cells with respective agonists for the MC4 receptor or any other GPCR that we're testing. We've just published these results in Cell. The paper came out in April 2019, and what we show in this paper is that we could measure both cyclic AMP and beta-arrestin interaction, and from comparing these two methods, we learned that actually measurement of beta-arrestin is much better predictor of body weight, and if we do the meta-regression analysis, the explanation for the body weight comes from beta-arrestin assay rather than cyclic AMP, which has been studied so far as the primary readout, primary outcome of MC4R signaling. Currently, we're working on a follow-up paper from this previous story. We try to understand any factors or any other properties of the receptor, especially carried by the variants of MC4R, which could explain these differences in the interaction with beta-arrestin. Other mechanisms which can underpin the body weight regulation. We're looking at other properties of GPCRs using the same NanoBiT system. Our research has wider implications. It first of all teaches us a lot about the biology, but second of all, it can influence the drug discovery efforts leading to development of new drugs. And this is our overarching aim. We want to help our patients, and the best help we can offer them is to understand their condition and come up with some mechanisms which can then be used in pharmaceutical industry, leading to development of new drugs, which can then help patients' life.